With microcirculation and Starling forces, microcirculation refers to the blood that flows through the smallest vessels in the circulatory system, called capillaries. And Starling forces, named after British physiologist Ernest Starling, also often called Starling pressures, are the forces that drive the exchange of fluid through the walls of the capillaries. The capillaries have a single layer of endothelial cells lining their walls with clefts between these cells. Normally, blood flows into smaller and smaller arteries, eventually reaching the arterioles, the medarterioles, and then the capillaries. In the capillary bed, due to the capillaries' thin walls and clefts, substances like nutrients or waste products can move from the blood into surrounding tissues and vice versa. After the capillaries, blood moves into venules and finally into veins. Intertwined with these capillaries are the lymphatic capillaries, which return interstitial fluid and proteins to the vascular system. Lymphatic capillaries can also empty into larger lymphatic vessels and eventually into the thoracic duct, which empties lymphatic fluid directly into the large veins. So, arterioles, medarterioles, capillaries, venules, and lymphatic vessels together make up the microcirculation. Now, the arterioles that come before the capillaries act as floodgates, regulating blood flow into the capillaries. So if the arterioles constrict, the resistance increases, and if they dilate, the resistance decreases. Therefore, the arterioles generally determine total peripheral resistance, or the amount of resistance opposing blood flow. This means arterioles play a key role in regulating the blood flow to an organ. Additionally, there are two mechanisms that help them do their job: intrinsic and extrinsic control. Intrinsic control of blood flow is based on the level of metabolites in the surrounding tissue. For example, adenosine and carbon dioxide will cause nearby arterioles to dilate. Another type of intrinsic control is autoregulation, and it's when the flow of blood is kept steady against changing arterial pressure. So, a sudden drop of pressure can reduce the movement of blood towards an organ, but the arterioles autoregulate by dilating, which reduces resistance in order to maintain blood flow. Then, there's active hyperemia, and when an organ becomes more metabolically active, its perfusion goes up to meet the increased demand. For instance, when a jogger is running, the blood flow to the leg muscles go up. Now, with extrinsic control, it's based on the sympathetic nervous and endocrine systems, which can decrease or increase vascular smooth muscle contraction, constricting or dilating the arterioles. And moving along the microcirculation to the capillaries. Here, substances can cross the capillaries in three ways. There's simple diffusion, vesicular transport, and osmosis. But overall, the most common type is simple diffusion. Normally, some substances can diffuse through the clefts and between the endothelial cells, but only if they're water-soluble. So, molecules like ions, glucose, and amino acids can readily pass through these openings. But there are others, like proteins, that are too big to fit through these clefts, so they have to cross in little membrane bubbles called vesicles. The exceptions are the capillary walls in the kidney and intestines, which are fenestrated, meaning they have large pores that allow some proteins to cross unimpeded. Alternatively, lipid-soluble solutes and gases like oxygen and carbon dioxide can just diffuse across the capillary walls. Additionally, the rate of diffusion of water-soluble substances and lipid-soluble gases are not the same. It all comes down to the total surface area available for them to cross. So, water-soluble molecules like glucose are limited to the clefts, while something like oxygen can diffuse across any surface of the endothelial membranes. This is why oxygen can diffuse into tissues faster than glucose. Now, while all these exchanges are occurring along the capillaries, another substance, water, is also making its way across, specifically through the endothelial clefts. It generally occurs by osmosis. No, not the makers of this video you're watching, but the movement of water across a semi-permeable membrane from an area of low solute concentration to an area with high solute concentration. And the net movement of water is determined by Starling forces, sometimes called Starling pressures, named after British physiologist Ernest Starling, who formulated the Starling equation. And it goes something like this, where JV is fluid movement, KF is the filtration coefficient, PC is capillary hydrostatic pressure, PI is interstitial hydrostatic pressure, sigma is reflection coefficient, pi C is capillary oncotic pressure, and pi I is interstitial oncotic pressure. The equation simply states that there are two types of forces acting within and outside the capillaries, which determine fluid movement. Let's start with hydrostatic pressures, which is described as the pressure that is exerted by a fluid in an enclosed space. In this case, inside the capillary or the interstitial space. Here we have PC, or capillary hydrostatic pressure, which pushes fluid out of the capillaries. And then the PI, or interstitial hydrostatic pressure, which pushes fluids into the capillaries. Then there are the oncotic pressures, sometimes called colloid osmotic pressures. And it's caused by proteins like albumin, which pulls fluids towards them. Here we have the pi C, or capillary oncotic pressure, which pulls fluids into the capillary. In addition to the pi I, or interstitial oncotic pressure, which pulls fluids into the interstitial space. But since there's usually little protein in the interstitial space, the interstitial oncotic pressure in normal conditions is close to 0. Next, let's look at KF, or filtration coefficient, which is not a force, but rather determines the water permeability of the capillary walls. It varies among different types of capillaries, and it depends on the size of the clefts or fenestrations. So, there are bigger fenestrations in glomerular capillaries, which let more water molecules through, and as a result, it has a high filtration coefficient. Cerebral capillaries, in comparison, have no fenestrations, so they have a low filtration coefficient. Lastly, there's the sigma, or reflection coefficient, and it describes the capillary's permeability to proteins. A coefficient of 1 means the capillary is impermeable to protein, whereas a 0 means it is completely permeable. So, when the reflection coefficient is 1, it means that plasma proteins, like albumin, are super concentrated in capillary blood and exert a great osmotic pressure. So, an increase in capillary protein concentration will lead to increases in capillary oncotic pressure, and therefore decrease the movement of water towards the interstitial space. To determine if the fluid is going into or out of the capillaries, we can use a simplified version of the equation where we ignore KF and sigma. Let's say that we are measuring the net pressure in the arterial end of the capillary. The PC, or capillary hydrostatic pressure, is 37 millimeters of mercury, and PI, or interstitial hydrostatic pressure, is 1 millimeter of mercury. Moving along, pi C, or capillary oncotic pressure, is 50 millimeters of mercury, while pi I, or interstitial oncotic pressure, is 0 millimeters of mercury. We get a net pressure of negative 14 millimeters of mercury. And since the number's negative, it means water will move into the capillary. If the number's positive, it means the water will move out. There are many conditions that alter these variables, so as an example, let's look at some causes of edema. In heart failure, blood can't get pumped out efficiently, so it backs up into the capillaries. This increases capillary hydrostatic pressure, PC, causing fluid to leak out, resulting in edema in the lower limbs. When there's an infection, toxins, or burns, it increases the filtration coefficient, KF. The clefts become bigger, letting more water through, which also leads to edema. What's more, when there's lymphatic blockage like filariasis, proteins leak out into the interstitium, which increases interstitial oncotic pressure, pi I, causing lymphedema. In other instances, like in nephrotic syndrome, liver failure, and protein malnutrition, there's a decrease in plasma protein, which decreases capillary oncotic pressure, pi C, and this also causes edema. All right, as a quick recap, The microcirculation is made up of the arterioles, medarterioles, capillaries, venules, and lymphatic vessels. And it's in the capillaries where the exchange of substances happen. The exchanges can be through simple diffusion, vesicular transport, and osmosis. Water crosses the capillary clefts with the use of the Starling forces. And the four Starling forces are hydrostatic pressure in the capillary, hydrostatic pressure in the interstitium, oncotic pressure in the capillary, and oncotic pressure in the interstitium. When the net force is positive, water moves out of the capillaries, and when it's negative, water moves into the capillaries.
Pressure is force over an area. So with blood pressure, we're measuring the force that blood exerts on the surface area of the walls of the blood vessels. Differences in blood pressure throughout the body keep blood flowing from high pressure areas, like the arteries, to low pressure areas, like the veins. When we say blood flow, we're referring to the volume of blood that flows through a vessel or organ over some period of time. Now, that amount of blood flow from one end of the blood vessel to another is affected by the blood pressure, as well as the resistance, which comes from the vessels themselves. Vasoconstriction, where the vessels constrict and get smaller, decreases blood flow. And vasodilation, where the blood vessels expand, increases blood flow. Now, blood flow is not the same thing as velocity of blood. Blood flow is the volume of blood that moves by a point over some period of time. So let's say that this chunk of blood has a volume of 83 cubic centimeters. And it took 1 second for this much to flow past the blue circle. This would be the blood flow, represented by the variable capital Q. Now, velocity, on the other hand, is the distance traveled in a certain amount of time. So maybe in that same 1 second, a red blood cell at the very edge here traveled a distance of 27 centimeters, meaning it would be moving 27 centimeters per second, represented by a lowercase v. Even though these aren't equal, they are related. And the last piece of this puzzle is area, specifically the cross-sectional area of the blood vessel, which in reality is the same as the blood cross-section, so it should look a little more like this. So based on units, since area is going to be expressed in centimeters squared, we see that the flow rate equals area times velocity. Alright, so as an example, let's say that we want to calculate blood velocity, and we already have a person's cardiac output of 5 liters per minute, which is average for an adult, as well as the diameter of their aorta, which is 2 centimeters. First off, using the equation for the area of a circle, which is diameter over 2 squared times pi, we get 2 centimeters divided by 2 squared times pi, which is 3.14 centimeters squared. Next, since cardiac output is the same as blood flow, we just need to convert this liters per minute to cubic centimeters per second. So there are 1,000 cubic centimeters in a liter and 60 seconds in a minute. So multiplying this all out, we get 83 cubic centimeters per second. Then if we rearrange our little formula, velocity equals flow rate divided by area, and we get about 26 centimeters per second, which also happens to be about 1 kilometer per hour. Alright, so getting back to blood pressure, blood flow, and resistance, that relationship can be written out mathematically as well. So to start, you have an initial higher pressure at one end, as well as a final lower pressure at the other end. The difference between these pressures, or initial minus the final pressure, sometimes expressed as delta P, equals blood flow through that vessel multiplied by resistance. This can also be written as Q equals change in pressure over resistance. So for example, let's say that the blood vessel narrows, which increases the resistance. In order to keep the flow of blood to organs the same, the pressure difference has to increase, and this is typically what happens. This equation might look familiar to a similar equation where change in voltage V equals current I times resistance R, which is also known as Ohm's law, an equation used in electrical circuits. All right, but let's get back to pressure. Blood pressure is usually highest in a large artery like the brachial artery, which is where blood pressure is usually measured with the blood pressure cuff. And that's because at that point, that blood still needs to be able to push through all the smaller arterioles and capillaries, which means it's going to meet a lot of resistance. In an average adult, blood pressure is about 120 millimeters of mercury over 80 millimeters of mercury. This number might look like a fraction, but it's not. That first number, 120, is systolic pressure, which is the force that the blood exerts on the walls of the arteries during systole, which is when the heart contracts to pump blood through the body. And the second number, 80, is the diastolic pressure, which is the pressure on the walls of the arteries during diastole, when the heart relaxes and refills with blood between heartbeats. And that's why the first number is larger. The arteries are under more pressure when there's more force from the heart pushing on the blood. Now, the mean arterial pressure, or MAP, is the average pressure on the arteries during a complete cardiac cycle, including the systolic and the diastolic pressures. This guy can be calculated in a couple ways. The first is the quickest way, where we assume that about 1/3 of the cardiac cycle is spent in systole, and about 2/3 of the cardiac cycle is spent in diastole. And so we say that MAP equals 1/3 times systolic blood pressure plus 2/3 times diastolic blood pressure. Now, another measurement is the pulse pressure, which is the difference between the systolic pressure and the diastolic pressure. So in our example, it'd be 120 minus 80, or 40 millimeters of mercury. So if we do a tiny bit of arithmetic, we can say that MAP is equal to 1/3 times the pulse pressure plus diastolic pressure, which is exactly the same thing, but just written differently. Now, a completely different way to think about MAP is by using the relationship between blood pressure, blood flow, and resistance. So let's go back to our big artery. Say this time it's the aorta, the biggest of the arteries, coming right from the heart itself. That artery goes to the arterioles and capillaries, let's say representing the whole human body. And then the venous blood returns to the right atrium of the heart. So in this zoomed out view of everything, the initial pressure here represents the pressure in the arteries. In other words, the mean arterial pressure. And the final pressure over here is the pressure in the veins, or the central venous pressure, or CVP, in the vena cava right before they enter the right atrium. Looking back at our equation, pressure initial minus pressure final equals flow rate times resistance. The flow rate is the total blood flow through all of the blood vessels and organs, which is also called the cardiac output, and is measured in liters per minute. Pressure initial is also mean arterial pressure, and pressure final is central venous pressure. And finally, the combined resistance of all the blood vessels of the systemic circulation is called the systemic vascular resistance, or the SVR. So the equation is mean arterial pressure minus central venous pressure equals cardiac output times systemic vascular resistance. Now, central venous pressure is usually a small number, so it's often ignored, and the equation is simplified to mean arterial pressure equals cardiac output times systemic vascular resistance. Based on this relationship, you can see why an increase in resistance, for example, which is mostly caused by arterial vasoconstriction, will cause the blood pressure to rise. Alright, as a quick recap, blood pressure refers to the force that blood exerts on the blood vessel walls. Blood flow refers to the volume of blood that flows through the blood vessels, an organ, or the entire body over a certain amount of time. And resistance refers to the physical or mechanical pushback that blood gets from things like vasoconstriction. What's happening, guys? Thanks for watching. That was a video on blood pressure, blood flow, and blood resistance, which you'll probably notice is a little more of a physiology topic as opposed to pathology. We're going to start getting into physiology a little more here and there, which is fun because I like physiology a lot. You get to work with equations and math, which kind of brings me back to my engineering days. So thanks to Andrea for writing the script on this one and Rishi for editing the script. I did the illustration and the voiceover, and Yifan did the editing. So thanks, guys. It turned out really well, I think. And if you guys want to kind of do a deeper dive on this topic, you should definitely head over to osmosis.org and make an account. We have a bunch of quiz questions and flashcards and other resources to kind of help you study. So if you're interested, check that out. Otherwise, you can help support us by donating on Patreon and subscribing to our channel and following us on social media. Social media. All right, guys. See you later. Compliance, which is sometimes called capacitance or distensibility, refers to the ability of a blood vessel to respond to an increase in pressure by distending or swelling and increasing the volume of blood it can hold. Or on the flip side, with decreased pressure, a decrease in volume. The way that this applies to blood vessels is to remember that they're stretchable tubes, like rubber hoses, rather than lead pipes. So if the pressure increases, the walls of the tube can actually stretch out a bit to accommodate a larger volume. And exactly how much they stretch out depends on their compliance. Now, we can calculate a given blood vessel's compliance, C, by dividing the volume of blood, V, in milliliters, by the amount of pressure, P, in millimeters of mercury that the blood's experiencing. And so we measure compliance in milliliters over millimeters of mercury. So when we plot out volume as a function of pressure, the slope, or volume over pressure, is the compliance. If you plot for the veins, they have high compliance, meaning they're high volume, low pressure vessels. And even a small increase in pressure expands the volume a lot. The arteries, on the other hand, have low compliance and are low volume, high pressure vessels, meaning with the same amount of pressure, their volume doesn't expand as much. Furthermore, a hardened artery would be even less compliant and is kind of like a lead pipe. In other words, it takes an incredible amount of pressure to change the volume even a tiny bit. With that in mind, since veins are more compliant, the majority of blood in the body at any given time is in the veins, whereas less blood's in the thicker, less compliant arteries at any given time. Now, when the arteries harden due to arteriosclerosis, they become even less compliant over time, which means they can't hold as much blood volume at the same pressure. That volume of blood that it can't hold anymore has to go somewhere, so it ends up in the veins. In this situation, blood simply moves away from the higher pressure arteries to the area of lowest pressure, typically where the compliance is highest, like in the veins. Now, if compliance, or volume over pressure, is its tendency to stretch out with pressure, then its inverse would be its tendency to not stretch out with pressure. Or another way to think about it is its tendency to recoil back to its original shape, which is a concept known as its elastance. Now, both of these concepts are super relevant for large elastic arteries like the aorta, which literally comes right off of the heart. Now, first off, heart cells take ATP and use that chemical energy to contract during systole, turning the chemical energy into mechanical energy. This propels blood out into the aorta. Since it's moving blood, it has kinetic energy. But also, since it's exerting pressure on the walls of the vessel, it also has pressure energy, which is a form of potential energy. And the sum of these two is equal to the total energy E, which we'll represent by this little health bar thing, which is filled with kinetic energy in blue and pressure energy in purple. So let's just say that during this time, that pressure energy corresponds to a pressure in the vessels of 140 millimeters of mercury. After the heart contracts, it relaxes and fills with blood, which is called diastole. Now, during this time, blood's still moving a little bit and there's still pressure, but there's much less total energy. So if there's less pressure energy, then let's say that it corresponds to something like 50 millimeters of mercury. If we take the difference between the systolic and diastolic pressures, 140 minus 50, then we get 90 millimeters of mercury, which is actually called the pulse pressure. Alright, but in reality, even though the aorta is an artery and isn't very compliant, during systole it still stretches out with these high pressure contractions. And some of that volume is held in this distended space. Since the aorta is very elastic, the walls store some of that energy of contraction as elastic energy in the walls, which is another form of potential energy. Since this total energy has to be the same as it was before, that means that there's less pressure energy and kinetic energy to make way for that elastic energy. So less pressure energy means that that blood pressure isn't as high. Let's say around 120 millimeters of mercury instead. Now, during diastole, those highly elastic walls recoil or snap back, which moves that blood that was held during systole out into the circulatory system and releases that stored elastic energy, converting it back to pressure energy and kinetic energy. So now pressure energy gets added during diastole, which let's say yields a higher pressure of 60 millimeters of mercury. This means that the new pulse pressure is actually lower. 120 minus 60 is 60 millimeters of mercury. So with elastic arteries, the blood pressures are 120 over 60, with a difference or pulse pressure of 60 millimeters of mercury. And remember that in the first example without the elastic properties, it was 140 over 50, with a pulse pressure of 90. So here we see that these large elastic arteries, like the aorta, buffer or dampen the pulse pressure. And this whole phenomenon is known as the Windkessel effect. As a person ages, healthy, compliant arteries become stiff and non-compliant. And this results in blood vessels that behave a lot more like the non-elastic case, which means pulse pressure ultimately goes up. Alright, as a quick recap, compliance refers to the volume of blood a vessel can hold at a certain pressure, and this is related to distensibility. Arteries are low volume, high pressure vessels, and therefore have low compliance. And veins are high volume, low pressure vessels, meaning they have high compliance. Aged arteries become much less compliant, which leads to higher blood pressure in the elderly. When we talk about pressures in the cardiovascular system, we're talking about blood pressure. Pressures in different parts of the cardiovascular system aren't equal, and these differences in pressures keep the blood moving from high pressure areas leaving the heart, like the arteries, to low pressure areas like the veins. Actually, though, the pressure curve looks a little more like this, and fluctuates in the arteries depending on what part of the cardiac cycle it's in. These peaks correspond to systole, and these low points correspond to diastole. That being said, though, this original line is the average of these fluctuations, or the mean arterial pressure, sometimes just shortened to MAP. Now, since systole takes up about a third of a single cardiac cycle, and diastole takes up the remaining 2/3 of the cycle, we can calculate the mean arterial pressure at any time by the equation mean arterial pressure equals 1/3 times the systolic blood pressure plus 2/3 times the diastolic blood pressure. So, for example, if the systolic blood pressure is 120 millimeters of mercury, which is the unit we use for blood pressure, and the diastolic blood pressure is 80 millimeters of mercury, plugging the numbers in, this becomes 1/3 times 120 plus 2/3 times 80, or a mean arterial pressure of roughly 93 millimeters of mercury. Another value we can look at is the pulse pressure, which is the difference between the systolic and the diastolic blood pressure. In this scenario, the pulse pressure would be 120 minus 80, or 40 millimeters of mercury. If we do a little rearranging to this equation, we get systolic blood pressure equals diastolic blood pressure plus pulse pressure. And then substituting that into our last equation, we get mean arterial pressure equals 1/3 times the quantity, diastolic blood pressure plus pulse pressure, plus 2/3 the diastolic blood pressure. And then after distributing, we get the equation mean arterial pressure equals diastolic blood pressure plus 1/3 the pulse pressure. Now, looking at these fluctuations on the arterial side, there's a couple important things to notice. First of all, on the downswing of the curve, there's a sharp pressure drop followed by a rise again, forming what's called the dicrotic notch, or incisura. As blood is ejected out into the aorta, pressure rises really fast. And then as the heart starts to relax, a tiny amount of blood flows back into the ventricle, which causes the valve to snap shut and the pressure to fall. That snapping shut of the valve causes it to recoil back a little bit into the aorta, which causes a brief increase in the pressure of the aorta. And then finally, the pressure falls as the aortic valve settles. A second interesting thing to notice is that the pulse pressure in the large arteries downstream of the aorta is larger than in the aorta itself. There are a couple of reasons why this happens. One is that the pressure wave from the contraction of the heart travels a tiny bit faster than the blood itself. To help kind of understand this idea, think of the molecules and cells in the blood like Newton's cradle. And while they move together, they bump into each other and end up transmitting that pressure wave faster than the group can move as a whole, meaning that that pressure wave actually ends up adding to the pressure downstream, therefore making the pressure higher in the large arteries. A second reason is that the pressure waves bounce off branch points in the arteries, which cause them to reflect back and increase the pressure in the arteries even more. Alright, so it should seem a little weird that both the systolic pressure and the pulse pressure are higher in the downstream arteries, since we know that blood wants to move from a high pressure area to a low pressure area. But one thing to remember is that in the large arteries, the diastolic pressure is lower. And remember that it's the mean arterial pressure that drives blood flow. And this is mostly affected by diastolic pressure, which is highest in the aorta. As an example with some numbers, let's say that the aorta's systolic blood pressure is 115 and the diastolic blood pressure is 85, meaning pulse pressure is 30. That said, the mean arterial pressure ends up being 95 millimeters of mercury. Now, if the large artery's systolic blood pressure is 120, which is higher than the aorta, and diastolic blood pressure is 80, which is lower than the aorta, then the pulse pressure ends up being 40, which is also higher than the aorta. But we see that the mean arterial pressure ends up being 93 millimeters of mercury, which is actually lower than in the aorta like we'd expect. Alright, so as we move out of the arterial tree, the pressure gets lower and lower as the arteries branch out into smaller and smaller arteries, and then arterioles and then capillaries. As those vessels branch into smaller and smaller arterioles, resistance goes up. According to the equation, flow rate Q equals change in pressure, or pressure initial minus pressure final, divided by resistance. With increased resistance, the pressure final or downstream pressure must decrease, which increases the change in pressure and keeps the flow rate constant. Across the capillaries, the pressure drops from 30 to 10 millimeters of mercury. So the pressure drop is greater across the arterioles than the capillaries, even though the capillaries are even smaller, which means even more resistance. The pressure drop here is less though because there are so many capillaries that run in parallel to one another, and that helps to reduce the total resistance, since total resistance for vessels in parallel is less than the resistance in any one individual vessel. After the capillaries, blood goes into the venules and then the larger veins. At this point, the pressure drops aren't very big at all, because the blood vessels have a larger and larger radius and have a high compliance, and can expand to hold more blood. Finally, blood makes it to the vena cava, and pressure is roughly 4 millimeters of mercury, and the right atrial pressure is around 2 millimeters of mercury. Even though there are only small differences in pressure, it's still enough to move blood back to the heart, even when it's going against gravity. Now, various clinical situations can cause pressures in the system to go up or down, and that affects all of the downstream pressures as well. As an example, let's take a closer look at the normal aortic pressure curve, and then two situations that often alter aortic pressures. The first is arteriosclerosis, which is a chronic condition where the arteries become stiff and less compliant, kind of like a rubber hose turning into a lead pipe over time. The baseline diastolic pressure usually doesn't change, But the extra volume of blood that exerts pressure on the walls during systole results in enormous increases in systolic pressure because the walls are so stiff. The high systolic pressure with the unchanged diastolic pressure means that there's also an increased pulse pressure, and therefore an increased mean arterial pressure. The second example is aortic stenosis, which is a narrowing of the aortic valve which increases its resistance. With increased resistance, the change in pressure must also increase, which means there's a decrease in the pressure after the valve, or the final pressure. The diastolic pressure again remains unchanged, but there's a decreased systolic pressure, decreased pulse pressure, and therefore decreased mean arterial pressure. Alright, so this whole bit that we just talked about is the systemic circulation. But once blood gets to the right atrium, it enters into the right ventricle and the pulmonary circulation. So here in the pulmonary arteries, the pressure is 25 millimeters of mercury during systole and 8 millimeters of mercury during diastole, which means that the mean arterial pressure in the pulmonary artery is 25 times 1/3 plus 8 times 2/3 equals 14 millimeters of mercury. That pressure drops to about 10 millimeters of mercury in the capillaries, to 8 millimeters of mercury in the pulmonary vein, and then 2 to 5 millimeters of mercury in the left atrium. Alright, as a quick recap, vascular pressures drive blood flow because the blood always wants to move from high pressure to lower pressure. The reason that blood pressure falls as the blood moves away from the heart is that blood has to get through many blood vessels, each of which typically offers a little more resistance. In the arteries, there are also pulsations or pressure fluctuations, and they get larger in the downstream arteries and then disappear in the arterioles. But mean arterial pressure always falls as the blood goes downstream.